Welcome to this biology review video, where we'll cover the 5 most important multiple-choice questions for MDCAT 2025 about prokaryotes, specifically Archaea and Bacteria. Let's begin with question 1. Which of the following features is common to both Archaea and Bacteria? A. Presence of peptidoglycan in cell walls. B. Lack of membrane-bound organelles. C. Linear DNA molecules. Or D. Eukaryotic ribosomes. The correct answer is B. Lack of membrane-bound organelles. Both Archaea and Bacteria are prokaryotic cells, which means they don't have a true nucleus or other membrane-bound organelles that are found in eukaryotic cells. Let me explain why the other options are incorrect. Option A is wrong, because while peptidoglycan is found in bacterial cell walls, it's absent in archaeal cell walls. Option C is incorrect because prokaryotes typically have circular, not linear, DNA. And option D is wrong, because prokaryotes have prokaryotic ribosomes, not eukaryotic ones. Moving on to question 2. What is the key difference between the cell wall of bacteria and Archaea? A. Bacteria have cellulose. Archaea have chitin. B. Bacteria have peptidoglycan. Archaea lack peptidoglycan. C. Bacteria lack cell walls. Archaea have thick walls. Or D. Bacteria have silica. Archaea have peptidoglycan. The correct answer is B. Bacteria have peptidoglycan. Archaea lack peptidoglycan. Let's explore the structure of bacterial and archaeal cell walls.
See the difference for yourself. With consistent use, our lash serum helps enhance the natural growth cycle of your lashes, leading to visibly longer, fuller, and healthier-looking lashes.
Bacterial cell walls contain peptidoglycan, a mesh-like polymer consisting of sugars and amino acids. This gives bacterial cells rigidity and protection. Archaeal cell walls, on the other hand, lack peptidoglycan and instead have pseudopeptidoglycan or proteins as their cell wall material. This is a significant biochemical difference between these two domains of life and helps scientists classify prokaryotes into their appropriate groups. Question 3 asks, which structure helps bacteria in movement? A. Capsule. B. Pili. C. Flagella. Or D. Ribosome. The correct answer is C. Flagella. Bacterial flagella are long, whip-like structures that rotate like propellers, allowing the bacteria to swim through liquid environments. They help bacteria move toward nutrients or away from harmful substances. Let's see how flagella differ from other bacterial structures. The capsule is a protective layer surrounding some bacteria. Pili are thin, hair-like appendages involved in attachment and genetic material transfer. And ribosomes are cellular components involved in protein synthesis. Only flagella serve the primary function of locomotion. Now for question 4. Methanogens are classified under which group? A. Cyanobacteria. B. Eubacteria. C. Archaea. Or D. Proteobacteria. The correct answer is C. Archaea. Methanogens are a group of microorganisms that produce methane as a metabolic byproduct in anaerobic conditions. They are classified under the domain Archaea, not bacteria. These organisms are particularly interesting because they can survive in extreme environments like very hot springs, extremely salty lakes, or in the digestive tracts of animals, including humans and ruminants like cows. In fact, methanogens play a significant role in producing greenhouse gases through their metabolic activities in various ecosystems. Finally, question 5. Which of the following processes is used by bacteria for genetic recombination? A. Binary fission. B. Transduction. C. Budding. Or D. Endocytosis. The correct answer is B. Transduction. Transduction is a process of genetic recombination in bacteria where DNA is transferred from one bacterium to another via a virus called a bacteriophage. Let's clarify the other options. Binary fission is the asexual reproduction method used by bacteria to create 2 identical daughter cells. Budding is an asexual reproduction method where a new organism grows from an outgrowth or bud of the parent, common in yeast, but not bacteria. Endocytosis is a eukaryotic cell process of taking in materials by engulfing them, which bacteria cannot do due to their cell wall. That brings us to the end of our review of the 5 most important MCQs for the MDCAT 2025 biology exam regarding prokaryotes. Let's quickly summarize the key points. Both Archaea and Bacteria lack membrane-bound organelles. Bacterial cell walls contain peptidoglycan, while archaeal cell walls don't. Flagella are responsible for bacterial movement. Methanogens belong to the domain Archaea, And transduction is a method of genetic recombination in bacteria. I hope this video helps in your exam preparation. Good luck with your MDCAT 2025.
Dreaming of MBBS? I chose Stars Academy Lahore for MDCAT prep, and here's why. Chapter-wise tests with video discussions, quarter, half, and full book tests, topic-wise tests.
Stars Academy Lahore is one of the best institutes for MDCAT preparation.
If you want to prepare for the MDCAT, I highly recommend Stars Academy Lahore for their excellent coaching and supportive environment.
Topic-wise tests with detailed explanations. Even past papers are explained chapter-wise. Quick revision? It's all online and